Okay, so now that you understand what is autosomal linkage, we'll be looking at how to draw the genetic diagram for autosomal linkage. Alright, so we're going to use the example in the notes to look at how to identify whether the inheritance is autosomal linkage, how to analyze the question, and of course how to draw the diagram itself. Okay, so for this question, it is very clear lah. They told you that the two genes, right, coding for the two phenotypes of flower color and plant height, the two genes are found on the same chromosome, right? So this tells you that this must be autosomal linkage. Of course, our usual questions wouldn't be so clear. Okay, but anyway, there are other clues to tell us this is autosomal linkage, which we will look at later, right? So tall plants with red flowers crossed with short plants with white flowers. F1 are all tall and have red flowers. So this tells you that the tall and red are dominant traits, and the parent with dominant traits should be homozygous dominant, because all the F1 are tall and red. Okay. So this also tells you that the F1 is a double heterozygous, right? Because the other parent would be a double homozygous recessive, right? Also double homozygous dominant crossed with double homozygous recessive, you should have double heterozygous, which are all tall and have red flowers. Okay, so when this F1 is test crossed, the following F2 plants are obtained. If you look at the numbers, right, this number is very different from the usual dihybrid inheritance that you have looked at earlier, right? So you have these two very large numbers for the tall and red and short and white, which are the phenotypes of the parents, right? So you have two very large numbers for the parental phenotypes and two very small numbers for the recombinant phenotypes, right? So tall and red are the same as the parent, right? Tall and red. And then the short and white, same as the other parent, right? So the two parental phenotypes, large number, two recombinant phenotypes, small number. This is another clue to tell you that this is autosomal linkage. All right. So I have these questions at below the example, right? Comparing it to the normal Mendelian dihybrid when the two genes are found on different chromosomes. Okay, so as we have mentioned just now, right, the parental genotype should be double homozygous dominant and the other double homozygous recessive, right? If this is the normal Mendelian, so you don't draw the lines, right? For the normal Mendelian dihybrid, you just write the alleles like that. Okay, so the F1 are double heterozygous, as we have mentioned, and the F2 should be 1, 1, 1, 1. So obviously this is not 1, 1, 1, 1. So again, two large numbers, two small numbers. This tells you this cannot be the normal Mendelian dihybrid. So even if I don't give you the first sentence here, right, telling you that the two genes are found on the same chromosome, you should also know that this certainly cannot be the normal Mendelian dihybrid, right? That the two genes cannot be found on separate chromosomes. Okay? Right, so again, this is like a very telltale clue to tell you that this is autosomal linkage. So anyway, to draw the diagram, of course, first of all, you need to give a legend in terms of the alleles, right? Because the question never tells you, right, whether to use R or T or any other letters. Right? So when the question don't tells you, you must do your own legend. Also in this case, I choose to use R and T, right, for the flower color and the height. Right? So because this is autosomal linkage, as we have talked about earlier, right, the way we draw the genotype needs to have the two lines to show the two homologous chromosomes, right, the pair of homologous chromosomes. Okay, so double homozygous dominant and the double homozygous recessive, shown as you can see on the screen and in your notes as well. So meiosis occur, right, because it's homozygous, so only one type of gametes is formed from meiosis. Then the two gametes fuse, of course, to give the double heterozygote F1, which is all tall and red flowers, right? Then the question tells you this is test cross, right, with the F1. So test cross is with a double homozygous recessive. Okay, and after meiosis, right, the double homozygous recessive only form one type of gamete. The double heterozygous can form 4 types of gametes. But if you recall our first video, right, these 2 gametes with the original combination of alleles would be the parental gametes, and the 2 with the new combinations of alleles would be the recombinant gametes. Okay, so because of the fact that we have the recombinant gametes, right, crossing over surely has occurred in prophase 1. If not, there wouldn't be the recombinant alleles, recombinant gametes, right? Okay, so I'm going to do a simple Punnett square here to show how we get the F2 genotypes, and then we can match it to the F2 phenotype and the numbers that we saw right in the F2. Okay, so the 2 here, right, with the higher number, right, they comes from the fusion with the parental gametes. And from our first video recording, we remember that the parental gametes are more likely to be formed. Right, again, right, for autosomal linkage, one of the feature of autosomal linkage is that these 4 gametes, they are not of equal proportion, right? The parental gametes are more likely to be formed, and therefore there are more of them, and there are fewer recombinant gametes. So that corresponds to why there are a lot more parental phenotypes and fewer recombinant phenotypes, right? Because these 2 phenotypes comes from these 2 parental gametes, which are more likely to be formed during meiosis by the F1. All right, so these 2 are the parental phenotypes, same as our parent, right? Red and tall and white and short. And these 2 are the recombinant phenotypes, which are fewer in number. Okay. So a bit of things to take note of in this diagram, right? You realize there are a bit more explanation included, which are required, okay, when you draw your own diagram, right? So these are not like explanation that I put in the notes for you to understand, right? These are required in the diagram, because your diagram is supposed to show how, right, you get this, to explain the whole inheritance, right, from parents all the way to the F2, right? So all these like extra explanation in a sense, right, additional stuff are actually explanations, right, which you should show to tell the examiner that you understand how the parents, you know, give rise to the F2. All right, so you need to show that crossing over has occurred in prophase 1. That's why you have the recombinant gametes. And also, right, you need to group the gametes and the F2 phenotypes into parental or recombinant groups to explain why that there are more parental phenotypes in the F2 than the recombinant phenotypes. That's due to having more parental gametes than the recombinant gametes after meiosis by the F1. Okay, so all these extra in a sense, right, compared to the normal diagram, right, all this extra stuff are actually necessary as part of the explanation of the whole inheritance and the 2 crosses. Okay, so this is what you need to do, right, when you are drawing the genetic diagram for autosomal linkage.